I'll be talking about in the next 30 minutes about the impending paradigm shift in drug design and discovery, something sort of a transition from what Dr. Yadva talked and where we might be going in the future. I'm afraid that I came here to check my presentation and I asked for the slide changes. Somehow I designed my presentations in a different way, but I would see that I would do a justice here. In the meantime, if that madam comes who was here, she showed me that she has a slide changer. That will be nice for me because I'm not used to speaking standing at one place. The moment I say that impending paradigm shift in drug design and discovery, the first thing comes to our mind, why a shift? What is wrong with the drug design and discovery that we are doing today? That's the first question immediately which comes to our mind. Because in a way, if we say that the drug, diseases, suffering, humanity, they are synonyms. The moment humanity came, the diseases came, the suffering came, and the quest of man for looking for drugs that came along with that. So at that time, because everything was revolving around nature, so men went to nature. That was the first obvious reaction. When we say to nature, as Professor Singh said, we went to the forest because that was nearest, that was the most well understood by the human being. So by trial and error, we learned to use different concoctions, different parts of plants like fruits, berries, bark, whatever was available in forest, but simply by trial and error that this works, this doesn't work. So we started using those for medicinal purposes. And all the ancient civilizations, they have made important discoveries in the field. And at that time, what was there that the civilizations were developing in isolation. So we had different concoctions at different places. Yes, no problem. Thank you, sir. So because of the development and isolation, so they had different kind of herbal remedies, traditional medicines, folk medicines, whatever we say. And out of all those, you see the Indian and Chinese herbal remedies are most well known. They are most very well documented. And some of those traditional medicine, folk medicine, herbal remedies, even some spiritual herbology remedies, they are still being practiced widely. Like this Muti Ifa, that is still being practiced, which is a branch of spiritual herbology in Africa by more than 35 million people, which is quite a sizable number. So all these, although these therapies they developed in isolation, but the basic underlying principle of all these remedies was the doctrine of signatures. This simply means that God has specially marked everything, and the job of the human being was only to find out the purpose. What is the purpose? Like a famous example which is always given by in our families by our forefathers is that because walnut resembles our brain, so it is good for our brain. And so much so that now today we have books like Language of Plants, where so many things have been related to various organs, that this is good for retina and something is good for our liver. So that is the basic underlying principle, doctrine of signatures of all those ancient traditional remedies. But all those concoctions or the products that we got from the natural materials, from not only from the forest, we went to the marine, we went to the animal kingdom. So these were, sometimes they were very effective, but sometimes they were very toxic also. And that's why the Greeks coined a common term known as pharmakon, which means poison as well as a medicinal product. That means Greeks at that time, they knew that a poison can be a medicine and a medicine can be a poison. So till 15th century, all these remedies or traditional therapies, they were being spread just by the word of mouth. But with the invention of the printing press, the publication and circulation and communication grew manifold, and along with that publication and circulation, the use as well as misuse of these herbal remedies, that also grew manifold. So much so that in 1566, Paris Parliament had to ban tartar emetic. By 17th century, their usage was at its maximum, but along with this, you see, effective communication amongst the practitioners, they started the withdrawal of some of the preparations, therapeutic preparations, which were too toxic. By early 19th century, our science developed a little bit, and we reached a stage where we could isolate pure substances from the natural products. The idea at that time was only simple, that if we are using a concoction from a plant, let us say that we are taking the extract of neem leaves, then the extract of neem leaves in July would be different as compared to the extract of neem leaves in December and March and all that. So they thought that, okay, why not to have a consistent quality throughout the year? To identify the active principle of the concoction, and then we have that compound, so consistent quality throughout the year. That was the quest that led to the isolation of the pure substances. But surprisingly, at that time, surprisingly for those times, majority of those active principles, single compounds, which were isolated from those active concoctions, they were found to be too toxic. Of course, some of those like morphine and cocaine were found to be useful, and they were very widely prescribed. By the late 19th century, we started searching for less toxic medicines or less toxic compounds. We reached an era where now we can synthesize lead-like molecules, what we called as analogs. So that means we have a pharmacologically active compound, mostly from nature, and then we can synthesize lead-related compounds, or what we call as analogs, and with that, there was a birth of the rational approach to drug design. And Paul Ehrlich is rightly known as the father of modern chemotherapy, because he was the original proponent of the magic bullet concept, where he said that a drug, a therapeutic agent, is just like a magical bullet, which can go and seek out and destroy the disease in the body. He was awarded the Nobel Prize in 1908 for medicine, and in 1910, he made a first fully synthetic drug known as Selvasan 606, or arsphenamine. And this drug was quite effective for treating trypanosomiasis and syphilis. So, in the nutshell, I have wrapped up, I have given you an overview of that where we are today, that what kind of drugs we are getting and from where we are getting. That is what I say as today's drug discovery process, or conventional therapies, or yesterday's drug discovery process. So that means whatever drugs we have today, almost all of them are coming from the natural leads. Then we isolate the active principle, we go to the synthesis of the lead-like molecules, we go to animal tests, and finally we go to the clinics. So that means like whatever simple drugs that comes to our mind at chemists, like say aspirin, or ibuprofen, or diclofenac, all those are just small molecule based. So which were either they are plant derived, or they are analogs from the plant derived molecules. However, if we see that how these small molecule based drugs, then majority of these drugs, they act at the protein stage. So say for example, if we have a protein lump somewhere, you go to a doctor, he would say either I can remove it surgically, or we can give a drug which can just dry it out. So most of these drugs, they are acting at the protein stage. But we know that the body's natural genetic flow is from DNA to messenger RNA, and from RNA to the proteins. So that means even if we are treating this protein in any way, let us say removing surgically or by any other means, the supply of protein is not stopped. So that means there is a further supply of the disease causing protein which is continuous, even though we are treating at the protein stage. But today's drug discovery, or tomorrow's, or the drugs of the future, they are not going to be, in all probability, they are not going to be small molecule based. They are going to be based on say genomics or proteomics, or in simple terms I can say that they are going to be DNA RNA centric. And it's not hard to think or hard to believe that why they are going to be DNA RNA centric, because after all, DNA is the language of evolution, a very simple 4-letter language. And ever since the structure of DNA was elucidated in 1953 by Watson and Crick, there had been a revolution leading to the sequencing of the entire human genome. And this sequencing of the entire human genome, that opened a plethora of opportunities, a vast array of opportunities to target those disease markers. And some of those opportunities come in the sense of what we call as antisense or anti-gene technologies. The major difference between the small molecule based drug design and the antisense based drug design, that in antisense based drug design, we don't use small molecules as drugs. We use short DNAs, what we call as sometimes as modified DNAs. We also call them as artificial DNAs, or simply oligodeoxynucleotides, ODNs. So short DNAs, modified DNAs, those are basically the drug candidates rather than small molecule based. And the difference being, you see, as I said, the traditional drug, the small molecule based drug, it acts at the protein stage, but the supply of the disease causing protein is still going on. Whereas in the case of antisense drug design, what we do, if we identify the disease marker on the messenger RNA, that is that part of the messenger RNA which is responsible for the synthesis of the disease causing protein, then what we can do, we can simply take advantage of the Watson-Crick base pairing. We can make a short DNA which is complementary to only that part of the messenger RNA which is responsible for the synthesis of the disease causing protein. Once we identify it by Watson-Crick base pairing, we'll have a DNA which will go and selectively bind to that part of the messenger RNA which is responsible for the synthesis of disease causing protein, with the result that there is no more protein synthesis. The protein which is responsible for the symptoms or for the disease, that is no more being produced. And our body has several mechanisms that if the further synthesis of the disease causing protein is stopped, then our body can take care of it on its own. So when I say that a short DNA can go and bind to the messenger RNA, what do I mean? Let us say that this is the part of the messenger RNA which is responsible for the synthesis of the disease causing protein. Then based on what is the sequence, that means ATGC, the simple 4-letter language, we can make a complementary short DNA which is complementary exactly to this messenger RNA. Now when this DNA is put to the cell or in solution to the system, what happens that due to this Watson-Crick base pairing, this DNA can go and bind and form a DNA RNA double helix, and now there would be a double helix. So now when this double helix comes to the ribosome for the synthesis, protein synthesis, there is no protein synthesis. And at this stage, we measure what is the hybridization affinity of the natural DNA or the modified DNA. So what I say that when we do this experiment, it's pretty simple. We keep on measuring the UV. It's called a thermal melting experiment or a melting temperature. Mind it is not the melting temperature as we know for the organic compounds. It's only a UV melting that we keep this complementary DNA with the messenger RNA or the DNA, whatever our target is, and we keep on measuring the UV, and from the first derivative of this Tm curve, we get the melting temperature or thermal melting or the hybridization affinity. Say for example, that if this is a natural DNA which is targeting the natural messenger RNA, and if the hybridization affinity is, let us say, 35 degrees, then if my modified DNA has to go and selectively bind to this messenger RNA, then its hybridization affinity has to be higher than 35. So if it is higher than 35, my modified DNA or artificial DNA would go and selectively bind to the messenger RNA. If it is less than 35, then the natural DNA would beat my modified DNA. So that's the simple thing. So that means now the major difference is that instead of targeting the protein, we can target that part of the messenger RNA which is responsible for the synthesis of the disease-causing protein. And with the result, when we have a double-headed, double-helix DNA RNA, ribosome doesn't recognize it and would not synthesize the disease-causing protein. So these short DNAs as drugs, they have several advantages over small molecule-based drugs. First of all, the specificity, because they would target only the single gene product along which it is complementary. Then the broad applicability, because we can use this technique or the technology to drug or to target those diseases which today we say are incurable or undruggable. Then the drug design is much more rational, because now the building blocks are just like Lego building blocks, the same building blocks. It is only the order which is being changed. So the drug design is much more rational, because it is not the different molecule, it is only the order. That means if I say ATTGC is one drug, then ATGGC can be another drug. So it is just like playing with the Lego building blocks, just having different building blocks at a different order, and we'll have a new drug. And then it is quite effective at the drug discovery and early development stage. And because we are dealing with only one kind of molecules, that is the nucleosides, so then the manufacturing can also be automated, because we don't need so many different kinds of reactors for different kinds of reactions. So historically, now it seems like that it's a pretty new concept, but historically it is not a new idea. The proof of concept was given by Zemachnik and Stephenson way back in 1978. But for the next about 30 years, this area stayed almost dormant. However, after the sequencing of the genome, because now we know so many genetic carriers, so many disease markers at the gene stage, so suddenly there is a spurt of activity. And in 1998, we got the first drug by the ISIS Pharmaceuticals. ISIS Pharmaceuticals USA is the largest company working in this area. They are working, if I remember correctly, since 1989. But that ISIS stimulates that part of the brain which the company didn't like. So after so many years, in February 2016, they changed their name from ISIS Pharmaceuticals to Ionis Pharmaceuticals. Anyway, that's with the name, but they are the largest company. So in August 1998, FDA approved the first drug based on this this approach, and that was what we call as the formivirsan or vitravine, which is an orphan drug for AIDS-related cytomegalovirus retinitis. Now, to give you an idea about how do you perceive the drugs today, like in your mind, if you have a picture of, let's say, aspirin or ibuprofen or diclofenac, so look at what is the structure of this vitravine. So it's a short DNA, it is a 21-mer. So it's a short DNA, you see GCGTT, that's what I'm saying. If I change this order, like instead of GCGT, if I have GTGC, it would be a different sequence and would be a different candidate as a drug. So this is vitravine. So all drugs based on this technology would look like this. Only the number can vary. This is a 21-mer, there can be 15-mer, there can be 49-mer. So it would only be a short length of DNA which would be complementary specifically to that part of the messenger RNA which is responsible for the synthesis of the disease-causing protein. Today we have about 140 clinical trials going on, including phase 2, phase 3 as well as phase 4 clinical trials. A majority of these trials are in the USA, followed by Europe and Canada. And potentially the clinical trials are going in those areas which today we consider as undruggable, like diabetes, cancer, cardiovascular, inflammation, apoptosis, antivirals, all those areas which today we consider that these are undruggable, these are incurable. The idea looks pretty simple, that we have a messenger RNA, we just make a complementary DNA and that would just bind and silence the gene at that place and would not let the disease-causing synthesis of the disease-causing protein. However, there are several challenges, like first of all, we need to identify the appropriate gene target. That's the main reason why for about 30 years this area stayed dormant, because we didn't know about these gene targets. Then we need to generate the antisense oligonucleotide or DNA which is specific for the gene of interest. And then whatever DNA we design, it cannot be a natural DNA, because the natural DNA, like we cannot take insulin because that would be degraded. Similarly, if we take a natural DNA, it would not act as a drug because that would be degraded by the nucleases, RNases or other enzymes. And then the drugs, whatever we need, they need to be deliverable to the organ of interest. That means the cellular uptake is another big issue. So that means because we need a stable duplex between DNA and messenger RNA, we need nuclease resistance, we need good cellular uptake, so we cannot use natural nucleosides. We cannot use the normal nucleoside ATGC that we get. That means the need of the time is Or if we have to go ahead with this technology, with this strategy, the need of the time is that we need modified orients, modified DNAs, or modified DNA monomers. And when it comes to modification of the DNA, there are 3' sites. We can modify the base, we can modify the sugar, or we can modify the backbone. And all these modifications that have been carried out extensively. Here are some of the modifications those have been done by our group, like 3' hydroxymethyl-LNA, or a beta-xylo or alpha-L-ribo bicyclic nucleoside. Then this is a dinucleotide which has been developed specifically for targeting the secondary nucleic acid structures. And recently we moved to the area of double-headed nucleosides. The term double-headed simply means that in a nucleoside we say that this is one head, nucleobase is one head. So when we say double-headed, if we have 2 nucleobases on one sugar unit, that is a double-headed nucleoside. And we really struggled a lot to publish in this area, because initially we will always get, the referee would always say what you would do with the 2 heads. But we have got now quite a lot of success in this area. We developed the spironucleoside also. Now we can put the second head anywhere, like for example the T is at the C2' of the sugar, or A is here at the C2' of the sugar. Or similarly, we moved into the started working into double coding nucleic acids, where the nucleic acid don't have to unfold to give its information and can give it as such. And recently we went into the arabino configured double-headed nucleosides, and the first review in this area is published by our group this year only, Australian Journal of Chemistry 2016. So today we know that we can put anything that is a second head in the major groove or the minor groove of the duplex wherever we need. We know that at what position it will go to the major groove and at what position it will go to the minor groove. So our interest was obviously to increase the stability of the duplex. Then whatever we make, it has to be nucleus stable, and most importantly, my DNA should go and efficiently recognize the messenger RNA without looking towards the DNA, because there can be a complementary DNA, there can be a complementary messenger RNA. So we developed our first molecule, and once again, if I tell you, let us say that this is the natural target sequence, 5' sequence, and then this is the natural DNA which can complement with it. So the TM is 33.3 degrees. That means if I have a natural DNA and then my own drug, DNA drug, which is also natural, so then the TM is 33.3 degree. Hybridization affinity is 33.3 degrees. That means my modified DNA, if it has to work, then with DNA it should not be stabilizing. Its hybridization affinity with DNA should not be higher, but its hybridization affinity with messenger RNA should be higher than that. That's the basic premise. So we wanted to explore this idea, and we developed 3 molecules having a triazole phenyl, triazole phenol, and triazole benzene sulfonamide at the nucleobase. Now the synthesis of these monomers is pretty straightforward from nucleoside point of view. That simply we protect, you see, the primary hydroxyl group with the DNT group, followed by Sonogashira coupling. TMS group is cleaved by ammonia, followed by click chemistry, and we have the phenyl triazole. Once we have this DMT protected molecule, we can phosphorylate it, preparing the amidite that is specific for nucleoside chemistry, because this molecule, once we have this DMTR and the phosphorus here, it can go to the automated DNA synthesis. For my younger friends who think that designing DNA or synthesizing modified or artificial DNA is just like a science fiction, it's a pretty straightforward, simple machine. It's a microprocessor based machine where you simply type here what you want, A, T, G, G, C, whatever you want, and all the natural nucleosides, monomers, they are here in the bottles, so it keeps on synthesizing on its own. Only where we have to put our modifications, it will stop, and then we inject, we do a hand coupling, and we get the DNA whatever we need in a pretty straightforward way. So we had these 3 monomers, X, Y, and Z, with triazole phenyl, triazole phenol, and triazole benzene sulfonamide, and now we were ready to build it into the DNAs. We wanted to see that if we can target the messenger RNA in preference to the DNA. So first of all, we made the sequences. You see, these are the sequences. ON1 means oligonucleotide 1, ON2, ON3, and here at the bottom, if you are able to see, there is a target sequence. It is 5' D, that means it's a DNA. So it's a target sequence, and the unmodified reference duplex TM is 33 degrees. That means if I have T here instead of B, if I have T, thymidine here, so then my reference TM hybridization affinity is 33 degrees. DNA versus DNA duplex natural is 33 degrees Celsius. Now the moment we replace this one of this T with X, Y, or Z, we see a considerable decrease in duplex stability. You can see that we come down to 28 degrees. So that means from 33 we came to 28. That means that there is a destabilization of about 5 degrees. Now if we have 2 modifications, still there is a destabilization, but the destabilization is a little less pronounced. You can see, for example, from 33 we come to 29, so there is a difference of 4 degrees, but we have 2 modifications, so 4 divided by 2, so overall decrease is 2 degrees Celsius per modification. So it is destabilizing. When we have 4 incorporations, still you see it is not stabilizing, it is still destabilizing. So the first test is passed. That means our artificial DNA is not going and binding to the complementary DNA. Now coming to the messenger RNA, that is what we need. So DNA RNA duplexes, once again, here the TM is 31 degrees, the TM is 31 degrees. So you see one modification One modification is still destabilizing, but it is less destabilizing as compared to DNA. There it was about 5 degrees Celsius. Here it is maximum up to minus 2 degrees Celsius. Now going to 2 incorporations, the moment we have 2 incorporations, we started to have increased duplex stabilities. So you can see from 31 degree with 2 incorporations we can go up to 39. So that means an increase of 8 degrees Celsius. So 2 modifications, 8 divided by 2, 4 degrees Celsius per modification. But you see from 31 to 39 there is a big jump. That means if I have a natural DNA and my modified DNA with 2 incorporations, my modified DNA would not let the natural DNA bind to the messenger RNA. Having 3 or 4 incorporations, there is immense stabilization. You can see we can go up to 55.5 degrees Celsius. So that is about 24 degrees divided by 4, about 6 degree per modification. If we look at from another perspective, going from 1 to 2 modifications, there is an increase of about 9 degrees Celsius. Going from 2 to 3, again there is an increase of about 10 degrees Celsius. And going from 3 to 4, there is an increase of again of about 10 degrees Celsius. That's remarkable RNA recognition. That is what we needed. That means no DNA stabilization, but DNA RNA stabilization. Just to see that what's going on there, we went to the molecular modeling studies and we found that this is the triazole benzene, triazole phenol, triazole benzene sulfonamide. We found that in the first 2 cases, the stacking is not proper. They are moving a little bit away from each other, whereas in the case of benzene sulfonamide, there is perfect stacking, which is leading to this stabilization. So going by the natural practice, we tried to modify, you see the orientation, we tried to change the linker, and we found that one of the molecules, what we name as the EBNA, that was found to be a good substrate for DNA polymerases. Mind it, if these molecules have to pass as drugs, they have to be substrates of DNA polymerases, and it was found that fusion DNA polymerase can effectively take it in a polymerase chain reaction. So finally, these molecules were taken up by the ISIS Pharmaceuticals, that I said, that largest pharmaceutical company, and they tested these molecules in human Huntington patient fibroblasts. They tested these against the wild type human Huntington RNA, Huntington disease RNA, only 1 minute, sir, the wild type and the mutant type, and they found that this molecule, that means the DNA having R incorporation, can have up to 23-fold selectivity. That means the modified DNA does not target the wild type RNA, but it targets only the mutant Huntington RNA. This has been published in the Nucleic Acid Therapeutics. Furthermore, it was found that in the transgenic mouse model, it has been found that there is no apparent toxicity throughout the mouse brain, even after prolonged testing. Finally, I leave you with the famous quote from the book, The Language of Life by Francis Collins, which says that we are on the leading edge of a true revolution in medicine, one that promises to transform the traditional one-size-fits-all approach into a much more powerful strategy that considers each individual as unique. Although the scientific details to back up these broad claims are still evolving, the outline of a dramatic paradigm shift is coming into focus. You have to be ready to embrace this new world. In summary, I just tried to highlight that we are ready to have a paradigm shift in drug design and discovery, and the promise of antisense-based drug, it has moved away from the science fiction to the era of personalized medicine, which is now a reality in the near future. And the rapid development of this antisense technology would lead to finding cures or drugs against those diseases which today we consider as incurables. And benzene sulfonamide, that group has been put by our group for the first time in the major group or minor group. And it has been found that our molecules, when built up into DNA, they can target the mutant Huntington RNA in real Huntington disease human fibroblast cells with 23-fold selectivity against wild type. And in transgenic mouse model, they had not been found to be having any toxicity. Currently, these molecules are undergoing exon skipping studies against muscular dystrophy. In the end, I would like to thank my long-term collaborator, Professor Paul Nielsen from University of Southern Denmark, where I spent all my summers since 2003. Dr. R.N. Vidu, who did the molecular biology experiments at Australia. 2 of the students whose work I have presented today, Dr. Pawan Kumar, my namesake, who is currently a Marie Curie Fellow at Oxford University, UK, and Dr. Navneet Chandak. And I thank Indian Science Congress Association once again, and thank to all of you for a patient hearing.